Hello and welcome to the Talking Precision Medicine podcast. In this series, we sit down with experts on the application of AI and big data analytics in the drug discovery space. Our guests are innovators, business decision makers, and thought leaders at the intersection of data and therapeutics. We discuss the promise, practice, challenges, and myths of AI in precision medicine. This show is brought to you by Genialis, and Raphael, its CEO, is your host. Genialis is focused on data integration and predictive modeling of disease biology to help accelerate the discovery and de-risk the development of novel therapeutics. Today's news is dominated by the coronavirus and COVID-19 pandemic, rightfully so. But today we're bringing you a story of another class of viruses called Phage. Phage are viruses that find and kill bacteria, and Felix is a Bay Area biotech harnessing the power of Phage to fight the scourge of antibiotic-resistant infections. So join our conversation with founder and CEO Dr. Rob McBride from wherever you have socially distanced yourself and learn about the viruses that are actually saving lives.
Thank you for joining us, everyone. Today, my guest is Dr. Rob McBride. Rob is an old friend of mine, and we've sort of recently been able to reinvigorate our professional friendship through the biotechnology world in the Bay Area. Rob is founder and CEO, your CEO, Rob, of Felix Biotechnologies. Felix is a new phage-based therapeutics company. Rob, why don't you tell us what Felix is all about and why phage is the future?
Thanks for having me on, Rafe. It's great to touch base with you again. Felix is focused on helping patients who don't have any other options. And the way we do that is by deploying phage specifically to manage bacterial infections that are not responding to traditional antibiotics.
So for
why is phage the future? Sorry.
Yeah. So for listeners who don't know, let's start with this. Tell us what is phage?
Yeah, phage are viruses. These are viruses specifically that infect bacteria. So traditionally, viruses can infect eukaryotes or prokaryotes, so human-type cells or bacterial-type cells, and phage are a subset of viruses that exclusively infect and kill bacterial cells. These are, one of the most prolific sort of entities on the planet, right? There's a number, I think, 10 to the 31 phage are on the planet. You are exposed to them and interact with them every day. They're on your skin, they're in your gut, they're in your hair, they're in the air. So they're all over the place, and they're very good at killing bacteria. That's kind of what they've been designed to do. And their ability to be used and to help humans battle with bacterial infections has been known for a long time. I think they were first deployed therapeutically back in 1919 by Felix de Hurel, who's actually, after whom we named the company, who also was a professor at Yale for a little bit of time. Back in 1919, obviously, we didn't know a lot of molecular biology. We didn't have sequencing. So there wasn't a lot of an ability to control phage and to deploy them in a controlled and effective manner. And so the outcome of phage treatments back in that time was not that effective. And so, in 1935, with the discovery of sulfa drugs, and then 1942 with the broad adoption of penicillin and the sort of golden age of antibiotics that followed after that, at least in the West, phage, were not really focused on as a tool for treating bacterial infections. And then I would say in the last 10 or 15 years, the rise of, the increasing rise of resistance to traditional antibiotics. So that's one of the factors that have driven the an interest in phage. A second factor that I think has driven the resurgence and an interest in phage is the understanding that, these antibiotics, which for the most part are broad spectrum, and in addition to killing the bacteria that's causing the issue, also kill a lot of good bacteria that are associated with positive health outcomes, that is not ideal, right? So people are interested in looking for more narrow spectrum solutions, and phage is obviously very narrow spectrum. Those are the two key factors that have brought, sort of more interest into it. And then, technologically, we are now in a place with regards to our molecular tools, our ability to sort of do synthetic biology and our ability to sequence things very cheaply, where we are in a position that we can deploy phage therapy, in my opinion, in an effective way and in a way that, helps patients who don't have really any other options.
I have a lot of directions I'd like to go in with that, but let's talk a little bit about the synthetic biology. So my familiarity with phage, since my academic background is molecular biology, is a tool in the lab. We used to use phage for either phage display of proteins or for various cloning techniques. What has changed, or what What is the synthetic biology toolkit that allows you to now harness phage and direct them for therapeutic purposes? And how is that maybe harder or more challenging than simply using them as a blunt axe in the lab?
Yeah, that's a great question. So the way we think about phage, right? Again, these are viruses. These are sort of a protein vehicle, right? So it's a protein, either a circle, it's got a tail on it, and it carries something, right? And most phage, wild phage, what they carry is sort of the code to replicate themselves inside a host, right? And so that replication, it comes along with the associated cost of death. So in a default situation, you have this protein vehicle that has the ability to find some target on a host. And so that's one element. And then the second element is what that protein vehicle is carrying. And so the default situation is that you find a phage that kills the bacteria, right? So that phage has a key to find that bacteria. So it hones in on that bacteria. And the second thing is once it finds that bacteria, it delivers the payload as death. And so those are the two elements that we think about as we think about deploying technology to help us harness the power of phage to manage bacterial infections, right? And so as far as the capsule goes, there's, again, two factors there. The one is, how big a payload can you put into it? So what's the size? And the second thing is, how versatile is your ability to target that, right? So one of the challenges with phage is that it's very narrow spectrum. And so, if you have 100 species of Pseudomonas that, are sort of isolated from, different patients' sort of, lungs who have Pseudomonas infections, you may have a phage that can infect, 20 of them, right? So it's very hard to find a phage that has broad host range. And so as far as engineering and synthetic biology comes into play, having the ability to take a phage that infects a subset of your target bacteria and engineer it with the ability to tune host range, right? To be able to infect more bacteria than it could originally. That's a huge advantage of sort of the current set of tools that we have. And I would say that area, that field is very nascent. So we can do that pretty well for a smaller subset. And we're working on being able to do that more broadly, right? Because the ultimate goal is to have a vehicle that you can direct at any cell type, right? So that would be the ultimate goal. And then as far as the vehicle goes, having right now the sort of the default for us is death, right? And amplification of the therapy. But we are working on, in collaboration with folks who know more about this than us, honing what we deliver, right? So we could, deliver death on the one end. On the other end, we can deliver things that, modify the DNA of the cell that we're trying to get into, or give it the ability, so not just fix or modify the DNA, but also put in new genes that give the bacteria or the cell that we're targeting the ability to make new things. And so those are the two elements, and our technology is focused on moving the fields in both of those elements forward.
There's a lot there again. I mean, there's a lot of work to do on payload engineering and specificity engineering and so forth. Where are you starting from? What did Felix start with in terms of kind of the founding idea or the founding thesis? And how do you think about prioritizing your engineering effort?
It's a great question, and I think the original idea for sort of deploying phage with a new flavor is originated in the lab of Paul Turner, right? And so Paul Turner is a professor at Yale. I did my postdoc with him, and he's got a great team assembled there now to sort of like take his initial ideas and turn them into things that are adding value to patients' lives. So the initial idea is, phage can bind. Pretty much, you can find phage to bind to any receptor on a cell, right, for the most part. And, when you do that, you kill the bacteria, and that's great. That's kind of what you want to do. But the bacteria is very good at evolving resistance to the phage. And so very quickly, a bacteria can become resistant to a phage, and then you're sort of, back to square one, right? So now the bacteria doesn't respond to antibiotics, and it doesn't respond to the phage. And so the traditional approach to managing that challenge is just to find, 2 or 3 or 4 phage to kill that bacteria at the same time. And what that does is it makes it harder for the bacteria to become resistant to the phage. And so what you're doing essentially is you're kicking the can down the road, and you're just delaying the evolution of resistance, right? But when that bacteria eventually becomes resistant to all the phage, now you're again out of luck, right? So you're in a situation where none of the phage work and none of the antibiotics work. And so that's increasing. So the outcome of an evolution of resistance is a bacteria that has more virulence. And so what really is unique about the Felix approach is we are really good at targeting phage to virulence factors. So these are things or elements that the bacteria has that makes them more harmful. And why we do that is really interesting. So if you target a virulence factor, the way for the bacteria to become resistant is to reduce virulence, right? So it trades off, resistance trades off with virulence. And so our approach is to ensure that, when resistance happens, because resistance is going to happen, it's inevitable, that resistance trades off with something that we're interested in trading off on. And so we don't want bacteria that are harder to treat after they become resistant. We want them to become sort of easier to treat.
No, that's fascinating. It's almost like saying, okay, you can evolve to stay here, but you got to quit being a jerk.
Exactly.
Quit hurting the patients. That's really fascinating. And what kind of validations do you have that this works? Has this been tried in patients yet?
Yes, this has been deployed in, sort of like at the bench. So we've done lab experiments to validate this. We have some mouse data to validate this. But the thing that we're most excited about is the technology that we are licensing from Yale has been deployed on a compassionate use basis to help patients with cystic fibrosis who have Pseudomonas aeruginosa infections that are treatment resistant, right? So they don't respond to any other antibiotics. And so in that context, when the physician who is managing those patients says that we've tried everything that we have and nothing works, the FDA will give permission to try some experimental therapies. And so we've been fortunate enough to be able to help now 10 patients with this approach with our lead asset. And in all cases, we've seen that finds the right bacteria. It kills those bacteria, so it reduces the bacterial load. It's safely deployed, so we haven't had any adverse safety events. And very important to us, when resistance to the bacteria phage, to our therapy happens, the bacteria then become resensitized to antibiotics because we're deploying our bacteriophage to target the virulence factors that drive resistance to antibiotics. And so we see, the bacteria becoming resensitized. And those are bacteria, once they're resensitized to antibiotics, then the patient has more options, right? And so that's ultimately our goal, is to try to deploy things that give the patient more options in the long run. So we're very excited and, motivated by the improvement in the quality of patients' lives that we've been able to help out with by deploying this therapy on a compassionate use basis.
That's fantastic. So I don't know much about the FDA mechanisms for this. Can you tell us a little more how these early compassionate use data impact then your clinical development plan? Like, can you steam ahead and start actually planning for larger trials, or is there some reason to kind of take it slow and work on optimizing the phage first?
Yeah. So again, another great question. Our philosophy here at Felix is, number one, the base of our pyramid is safety. The second layer is good science, and then after that is like patient value, and then after that is sort of commercial value. So that's kind of our Maslow's hierarchy. And so the first thing, obviously, is safety. The FDA's perspective on compassionate use is that data is, the goal of that use is not to collect data, right? So we're not doing compassionate use studies to collect data to help us advance the commercial prospects of a therapy at all, right? We're just trying to help the patient, and that's the focus. In so doing, we do get some information that is helpful to us, but we are in the process of making sure that as we move this therapy forward to be able to be accessed by more people than just those who are out of options, we do it with a safety first metric, and then after that, quality of science, and then after that, sort of utility for the patient. So the next step for us is we are in the process of moving forward with a research IND. The technology is being evaluated using a research IND. So this is the output of this activity would not be the rights to market a drug. The output of this activity is to collect data to understand more about the drug. And it's a small trial. It's a 3-patient trial, and we're primarily focused on safety and also understanding more about the dose. And so that's kind of the next step for the company, is to just get more data, controlled data regarding the safety and sort of the appropriate dose. And so, that in conjunction with our preclinical data and information on how we make this commercially would then go into our application for, doing a commercial trial, which would be a larger trial with more controls. So that's kind of the path.
I'm curious a little bit about the safety risk profile. Is the potential risk due to, some sort of host immunogenic event, or is it some, off-target consequence or off-target killing, or is it a consequence that you can't predict of actually hitting the target, but something bad happens? Or is it really just we don't know, so we have to be careful?
Yeah. So, again, when you're dealing with sort of humans or any sort of like living thing, even if it's plant. I think the first thing is, do no harm, right? And so phage have been deployed in humans for more than 100 years now. And, as I said, in the West, we didn't think about using them that much from the 40s through the, maybe the late 90s. But, phage have been used throughout that time frame in sort of Eastern Europe and a lot of those countries. And so to date, as far as I'm aware, there have not been any adverse safety events associated with the deployment of phage, right? So phage have been deployed through inhalation, right. Phage have been deployed through IV, they've been deployed topically. So there isn't any information to date to suggest that there could be, that phage are not safe, but we move forward with an abundance of caution, right? And I think the biggest primary concern, and there's not a lot of data to support this concern, but it's a protein, right? And if you're deploying it intravenously, your immune system recognizes this, and so you've got the innate and the adaptive, and so there's a lot of things that could happen. So for us, we're focusing on outpatient therapy, but the first application always happens in patients so that we can monitor for things like allergic reactions.
Sure, that makes a lot of sense. And I imagine dosing's not trivial. You probably have to, since it's a biological system, the PK/PD considerations are going to be somewhat different than a small molecule or a chemical that you're administering.
Yeah, not just a biological, but it's an amplifying biological, right? So it makes it very, very challenging. So that's an accurate statement.
Are there well worked out strategies for thinking about dose and thinking about measuring that amplification?
Not that I'm aware of. I think a lot of folks are moving this forward, and as are we. We're learning as we're moving this forward, and the key thing for us is let the science drive us, right? And so as there's a lot of variability in humans that you don't see in sort of animal models, and animal models that you don't see in lab models. And so for us, the key thing is just take this one step at a time, check all the boxes, make sure being as safe as possible, get as much data as we can, and then leverage that to, again, try to get this into the clinic to help patients more broadly.
Yeah, that makes a lot of sense. I'm going to circle back a little bit to the synthetic biology. So in a previous lifetime, I worked in a synthetic biology group. In fact, one of my colleagues, just a couple benches over, is one of your co-founders at Felix. I'm talking, of course, about Vivek. And what interests me about this is you have the potential not only to have a really important therapy for seriously like society-threatening diseases, but also a platform technology, the ability to kind of engineer these things and tweak the targets and the underlying function in a very high throughput way is appealing to me. How do you think about balancing your activities on we've got to get a drug into the clinic versus we want to build a cool platform that we could point everywhere?
And I think our philosophy is that the technology is in service of helping patients, right? And so that's kind of whenever there's a question, we use that as our guide, right? Is this development helping us make something that's going to improve someone's life? You're right. We've got a number of technologies actually that we are working on that have originated in Berkeley. The first is the targeting technology, and Vivek just had a paper come out on that, which is really exciting for us to get that out and to have people be able to see how powerful and impressive this tool is. And essentially, the first piece of technology really allows us to, without needing to engineer a phage to target any host factor, right? We can find the right phage for a particular factor, more quickly for less money than any other technology that we're aware of. We're not at the point yet where we can engineer a bacteriophage to target any target we want. And so in the meantime, we can use our technology to find the right phage to target any target we want, right? So that's kind of step one. Step two would be engineer small host ranges, which we can already do to some degree, and we'd like to increase our ability to do that moving forward. And then step three is, be able to target anything, right? And that may be one vehicle, that may be a few vehicles, but that's definitely sort of the ultimate end goal, is to have this universal vehicle that you can aim at any target on any cell type, right? And so that's a pipe dream that's down the path. We're definitely not there yet, but our focus is just very, again, it's patient centric, right? What is the problem we're addressing? How are we helping these patients, and how can we deploy this technology most effectively to do that? Obviously, there's a lot we can do, and so we are open to working with other groups to leverage the technology to help other people and to help go after other targets. But with the resources that we have and the focus that we have, we've just got to, again, do the minimum that we need to do with the technology to get from point A to point B.
Yeah, no, that makes a lot of sense. And it seems like you put a good team together to get from A to B. Tell us more about the team. Who are you working with directly? Tell us more about some of your founding advisors and Felix family at large.
Absolutely. So you already mentioned Vivek Mutalik. Vivek is heading up the science. He's the CSO. He's a very humble scientist, as you know, but he definitely is one of, in my opinion, one of the top synthetic phage biologists in the country, and is, a co-inventor on a number of the technologies that we are working with. And so, not only creative, but hardworking and also, aligned with the vision. That's a key thing for us in our team, is you have people aligned on the vision and the mission and the values, have non-overlapping skill sets. And we think that positions us in a good place to have a chance at winning, right? So there's a lot of challenges, but that's something we need to do well. We have Natalie Marr, who's heading up our business development. She was at Yale too, worked in Paul Turner's labs for a while, so deep technical experience, and has been in healthcare consulting for a while now. Very passionate and an amazing addition to our team. We've got two scientists on the ground, Quinan Ma and Tanya Silvis, and they're amazing scientists, and they're helping to drive us forward on the ground. So that's kind of the on the ground team. As far as our advisors go, so Vivek is a co-founder, and then Paul Turner is a co-founder, and Adam Arkin's a co-founder. So those are two of our advisors. Paul's one of the sort of the leading folks in phage evolutionary biology, as well as phage therapy, National Academy of Science member, and just a wonderful co-founder. And, obviously I was in his lab, more than 10 years ago, and know him well, and have some good history with him. We're really excited that Adam joined us. Adam and I had teamed up on a previous company, and I was very aware of the capability and the extent of the technology that he had that could be brought to bear to help us do a better job helping patients. And he was very excited. And so he's obviously one of the leaders in the field of synthetic biology. So it's great to have him on the team. We've been very fortunate to have Jennifer Doudna join us as a scientific advisor as we move forward and think about deploying our vehicles as more sort of general payload delivery vehicles. Having her expertise, her sort of cell engineering and payload expertise on board to help us tackle some of the challenges that are limiting our ability to progress is priceless. So very excited to have her on the team. Adam Deutschbauer is another scientist who's helping us out, and he's perhaps the leader in the field of developing tools to interrogate functional genomics in high throughput for low cost, right? So he's spent a lot of time and a lot of his effort, building tools to do this quickly and cheaply. So that's more the technical advisors. And then as far as the clinical advisors go, we've got a cystic fibrosis physician at Yale, John Koff, who's sort of heading up the research IND and administering consenting patients for the compassionate use. Amazing, passionate, and wonderful to have that physician perspective on our team. Marie Egan's one of the leaders in the field of cystic fibrosis too, and very helpful and useful in helping direct us. Ben Chan, again, one of the top sort of applied phage biologists in the country. He is in Paul Turner's lab and has perhaps, single-handedly done more for the academic side of this field than anyone else. And so we're super excited that he's helping us out. And then we've got a young cystic fibrosis patient called Max Lebo, who has cystic fibrosis and actually reached out to us as he was looking to help companies develop tools to manage microbial infections in cystic fibrosis, because that ultimately ends up being one of the ways that most people with cystic fibrosis is sort of like, pass. Yeah. So that's kind of the team that we've got. We do have some holes that we're working on. So we're very focused on bringing in some folks with clinical experience in terms of bringing drugs through the FDA. So we're looking for a CMO type role right now. And we've got some great data science help right now, but we're looking to make that more permanent.
Makes a lot of sense. So anyone out there who's listening who is or knows of a great CMO in the space, Felix is hiring. I want to talk about two things, Rob, and these are maybe different sides of the same coin, but they're a bit at odds. The first thing I want to understand from you is just how big and how severe is the scale of the problem of untreatable bacterial infection? When I was last working on antibiotic discovery, which was probably 6 years ago, we had some stats, and it was kind of alarming, the number of multiple resistant infections that people get in hospitals. And this is not just a developing world problem. This is a first world problem. I was at the Texas Medical Center, arguably the most modern medical center in the world, and these things happened. So how big is the human problem, the health problem, the economic problem of untreatable infection?
It's alarming, right? The problem is alarming now, and the projections into the future are even more alarming. So right now, last year, so the World Health Organization and the CDC just recently both came out with reports, so this data is all easily accessible. In the US last year, more than 35,000 people died from bacterial infections that are resistant to antibiotics. So about, just under 3 million were diagnosed with infections that were resistant to sort of first and second line therapies. So that's just in the US, that's an amazing number to me, right? So these are infections that we thought we had managed and sort of licked back in, 1942 with the sort of broad use of penicillin, right? And so I grew up in an era where we didn't, I never paid attention to bacterial infections, right? You take antibiotics like you take sort of like headache medication. And, we're making a bet that we want a future that in which we don't go back to that period of time pre-1942, and that other people are also going to want a future in which bacterial infections are not once again one of the leading causes of death. So that's the US challenge, right? And globally, the number this year was closer to 700,000. And these are pretty conservative estimates. But the World Health Organization predicts that if we don't do anything, right, if new tools aren't developed, and not a lot of them are being developed, so that maybe is a point to discuss in a little bit, the market challenges here. But if new tools aren't developed by 2050, so that's, less than, it's about 30 years from now, these deaths so there will be about 10 million deaths a year from these infections. That is a number that's larger than the combined deaths attributable to cancer, right? So that'll be more deaths than cancer, and everyone knows somebody who's, been impacted by cancer. And so if we don't do anything, these infections are going to be more impactful than cancer. And so that's not a future that we want. And, we know that we're one of the solutions, and we're a very, specific solution, and that more solutions are going to be needed. And so we're just focusing on doing our part to help prevent that future from manifesting.
Those numbers are similar to what I remember, but I guess updated, and alarming is the right word. I mean, I like to joke, it's the little things that kill you, right? but in this case, in a very real sense, it's the bugs we thought we'd had. In a way, it reminds me of kind of the risk of, eschewing vaccination and allowing bugs to kind of rear their head again, except here we're almost, sort of over treating with antibiotics and, throwing them into our water supply and our produce in our agriculture, and kind of making it worse for ourselves. But given the clear health need and human need for these tools, you mentioned market challenges. That was the other side of the coin. That was the tension I wanted to get to. Why is it hard to basically to build these tools? Why is it hard to get people to pay for innovation here?
That's a great question. And that's the question that is really frustrating the field as a whole, right? And so, in 2019, I think 3 companies that were successfully developing new small molecules to manage bacterial infections went bankrupt, right? So, here locally in the Bay Area, Phage was perhaps the sort of the best known of those stories. And then another company, a large company, was Melinter. And so these are companies that have spent, upwards of $500 million, taken 10 years, managed all of the huge risks associated with developing a new molecule, right? So discovery, translation, preclinical, gone through clinical trials, had technical success in clinical trials, gone through the regulatory process, received permission to market. And so that is just a huge achievement, right? So they've had this amazing success, and then they get into the marketplace and they can't make money, right? So Phage, their first year sales was I forget the number exactly. It's somewhere around 3 million, I think. And and so they're like, that's not going to work out. We have to declare bankruptcy, right? And so it's, there's 2 primary reasons driving the sort of the revenue challenge. One is, how much you can charge, right? And so if you're treating something that's inpatient, you're subject to the constraints of this thing called the DRG, which limits how much you can charge. And then there's a program in the US, in sort of like a federal program, but then there's states and hospitals that all sort of are trying to implement this, called microbial stewardship. And the goal of that effort is to limit the spread of antibiotic resistance. And so what the stewardship programs do is they will manage and keep new exciting therapies that work as last line or third line options, right? So what that does is it reduces the volume, right? So when you're trying to make money, you can charge a little and sell a lot, or charge a lot and sell a little. But the constraints that are sort of in the marketplace today limit your ability to do either if you're successful. So the deck is stacked against us in terms of the market risks, which makes this a very challenging area, right? So most large pharma have gotten out of the space. Most companies in the space are not doing well financially. And, what that means for people who fund innovation is that they look at this and they say, this is not an area that we think is, we're not going to put a lot of dollars into this. And that's really been reflected in the amount of investment that's gone into new infectious disease companies versus the amount of investment that's gone into other areas. So huge challenge. in terms of what can be done, right? So there are some, bipartisan regulatory solutions that have been proposed that, haven't been passed yet, but they may do some interesting things in terms of adjusting the rewards, right, and making it more possible for companies to make money, sort of like post-approval.
Yeah.
But those haven't passed yet. And so it's a tough spot, right? It's a tough situation to be in. And if that continues, right, we're not going to have this innovation and this drive to develop these new solutions. And we're going to have a future that is, not a future to be excited about, which is not something we want here at Felix.
Yeah. I mean, healthcare pricing and healthcare economics is sticky and probably needs to be like its own podcast series unto itself. But, I'm just thinking about the kind of insane contrast with the recent breakthrough, seemingly curative gene therapies, but these are going at like $2 million a pop, right? And what you outlined for us is, a future where there are going to be 10 million deaths a year without the kinds of drugs that we're talking about. We're not talking about 10 patients globally. We're talking about millions, tens of millions. So, it's this sort of law of supply and demand seems to really be kind of shooting us in the foot here. Are there programs does the FDA have a voucher system like they do for certain rare diseases? Like, are there ways at least of making the initial risky bits cheaper or less financially risky?
Yeah, so it's a great question. So the FDA doesn't have vouchers yet, right? But I will say that, I've painted a pretty bleak picture, right? But I do think that there are ways to mitigate risk, right? Market risk. And so we are, the first thing we did as a company was sit down and look at this market challenge and convince ourselves, right? And we'll do the research to ask the question, if we are technically successful, right? So if all of these other technical challenges are managed, right? Is there a path to commercial viability? And that was very important for us because if we don't see a path to commercial viability before we do all the technical stuff, we shouldn't be doing this, right? We should be funding this non-dilutively. This should be a sort of like a compassionate use approach. And so for us, the solution is very simple, right? So if you can if you've spent $1 billion and taken 10 years to develop a drug, you've got to sell a lot of it to substantiate that investment, right? So one thing we can do is not structure ourselves like a traditional small molecule venture-backed company, right? And to be as creative as we can in terms of spending as little as possible to get to the point where we can start selling this, right? And so, one of the ways to do that is to go after an orphan drug designation, right? So in an orphan drug designation, you don't have to, there's not as many patients needed in the trials. You can get some tax benefits. So you can target an indication that allows you to develop something for less money and for less time, right? So that's one lever that we have, right? And so we're very focused on doing that and to try to bring in as much non-dilutive support as we can, because there's a lot of push incentives in the U.S. I'll call out CARB-X. It's done an amazing job. Nova Repair Funds does an amazing job, and there's a lot of support from government institutions to help non-dilutively support this. So that's one lever. And then as far as the market goes, right, the two levers you're playing with are pricing and volume, right? And so, pricing is primarily constrained in inpatient care, right? So we are designing our therapy to be an outpatient therapy where we've got a little bit more flexibility in pricing. And, when we look at the standard of care in the space that we're in, which is, Pseudomonas aeruginosa infections in cystic fibrosis patients, if we can price similarly to the current standard of care, and so that's going to be an important component of our financial success. And then the last point that we think is an important point to make is, our argument is that Phage should not be subject to the constraints of stewardship. Stewardship's goal is to mitigate the spread of resistance. We have a therapy that actually decreases the spread of resistance by converting things that are resistant to sensitivity, right? So we think there's a really good argument that Phage can be a first-line therapy. And so we think with those approaches and some assumptions, right, so you manage the amount you spend, and then once you've gotten into the market, you have flexibility on pricing, and you are not constrained by volume. And, we think that's the right recipe to take, at least to show people that there is a commercial viable option here. And once a few companies start to demonstrate that, I think that will open the floodgates and have more funding go into this space to develop more tools, which we're going to need as we move forward and antibiotic resistance becomes a bigger issue.
Yeah, I mean, it's an uphill challenge. You've got the scientific and clinical stuff to deal with, and of course these market forces. But it may be that we shouldn't let a good catastrophe go sort of untapped, and that the health challenge itself will force the market to adjust to this out of need.
I mean, and patients can't wait, right? That's the key thing here. So we've got to figure out a solution, because there's folks, dying every day who, we may be able to help if we move forward more quickly.
I do love that, and this is clear. It oozes from you. I love that Felix has such a sort of crystal clear North Star that, this is why, for those of you who don't work for companies or haven't kind of built companies before, this is why companies have values and have cultures. It's so it helps you make decisions, and it answers your questions for you. And your focus on what will benefit patients is missing from, I think, a lot of the heavily monetized drug discovery, where people are either more interested in the tech or the market opportunity than the patient need. That's not universally true, but your focus there is commendable. What's next? What's next for Felix? You're in Y Combinator. That's exciting. Today you're down at Illumina, so you're working with all the right people. What are your kind of milestones for 2020?
Currently fundraising. We have a seed note that we're looking to close out, and the seed note allows us to hit the milestones that get us to the point where we can start working on our commercial IND. And those are to wrap up some engineering on our lead assets, to do more preclinical safety work, and to support the research IND and get some information from that to allow us to hone our protocol for the commercial IND. So 2020 gets us to a point that we're ready to start working on the commercial application of it.
Fantastic. I'm looking forward to following your progress. We should have you back on once that part is underway. Rob McBride, thank you very much for joining us. I appreciate the conversation.
Thanks, Rafe. I really appreciate the opportunity, and great job on this podcast. It's been fun listening to some of the other podcasts, and please keep up the good work, because this is very interesting. So we appreciate it.
This has been episode 19 of Talking Precision Medicine.